Can you see the screen that I'm sharing, everyone? Is it visible to all of you?
Yes.
Okay, perfect. Okay, so we were actually figuring out the genetic material. And so who would like to give a quick recap of the search for genetic material, Griffith's principle and every MacLeod-McCarthy's experiment quickly in 5 minutes? Anyone would like to try? Give it a shot. So this exercise is also just for you so that you gain confidence and also concept. When you explain someone, concepts get strengthened in your own head. Yeah. So who would like to go? Anyone? Come on, volunteer. Quick. No, no one. Then I'll have to pick. Okay. So I'm sparing Alia for her throat. How about we go to Parker then? Parker, how are you? Would you like to give a quick summary? Am I audible to you, Parker? Come on, quick. I've got something in a chat box. Oh, Parker is also not keeping well with her throat. I think it's a season, I guess. Okay, so Alia, Aisha and Rabia are left. So anyone would like to volunteer, or let's go alphabetically? Aisha, would you like to give a quick recap? It will be great if you give it. And don't worry, you'll just discuss what we did and what you learned, and anything you miss out, I'll just pitch in. Are you there, Aisha?
Yes.
Would you like to do it?
Yes.
Okay, great. Let's do it.
So we study about the Fed's transforming principle. Then
Then you are mute, Aisha, if you're speaking.
yes, sorry.
Yeah.
So Griffith performed an experiment where he took a piece of corpus pneumonia and he injected two types of strain in the mice, S strain and R strain.
Right. What was the experiment quickly, and what did we learn from it?
When he injected the S strain, the mice developed pneumonia and died.
Yeah.
And
Right.
when the injected R strain and injected mice, so the mice lived.
Right. And what does this tell us about the baseline nature of these strains? That S strain is virulent and dangerous or lethal, whereas R strain is non-virulent and non-lethal, right?
Yes.
Yeah, continue.
Next, he killed the S strain bacteria and injected it along. It showed results wherein the mice lived. And in the next step, he took the S strain heat kill and R strain, which was live, and he injected in the mice, and the mice died. So he concluded that the R strain had inherited genetic material from the heat kill S strain bacteria, which made it very.
Right, which transformed it which made it a S strain bacteria, right? And he also
Yes.
proved it by isolating S strain later on from the mice in which he injected a R strain live and a S strain heat killed. He could recover live S strain from this mice after the death. And this proves that the R strain indeed got transformed into a S strain using some genetic material from the S dead heat killed S strain. Okay, so I think I lost the electricity, but it will be back in some time. Okay, and then what was this? What was the next experiment? Aisha? Good evening, Iram. Iram just joined in. Aisha, continue. Am I audible? People, am I audible?
Yes, sir.
Yes. Yes. Aisha, are you there? Have we lost Aisha? Connection? I guess, yeah, she's not. She'll be rejoining. Okay. Anyone would like to take up this, the biochemical characterization? Rabia? What was the next step? Aisha just joined in again. Okay, let her connect to the audio and then we can. Okay, in the meanwhile, she's connecting. I guess there's some issue. Okay, Iram, how are you? Are you there, Iram? Would you like to talk about this? Yes, yes, yes. Go ahead. Yes, Iram, you're audible now. And
Yes.
not, yeah. Would you like to talk about this? What every MacLeod and McCarthy did?
Yeah. Okay. So every McCall and McCarthy, they purified chemicals from heat, kill S strain. So what they did is to prove that DNA is the genetic material. They took test tube and added proteins, lipid saccharides, and nucleic acid to it. And for proteins, there was no transformation. For lipids, there were no transformation, and for saccharides also there were no transformation. But only with DNA there was, and it was like proof that DNA is the genetic material.
No. So what they did, so when you say this is what first was done, but the proof that DNA might be the genetic material is not when you inject it and it shows transformation. One can easily say that there might be contams present along with that. So what they did, they used digesting enzymes, right? They took the whole cell extract and first they put protease, so all proteins were gone, but still there was transformation. Then they put lipids, which means all the lipids were degraded, still there was transformation. In case of when they degraded all the DNA using DNase as an enzyme, there was no transformation. So the negative result where the transformation was not happening is a more robust proof to say that DNA is the genetic material. Instead of injecting just the DNA, which you are saying is DNA and is not contaminated and it shows transformation. Both actually prove the same thing, but the absence of transformation, because transformation was anyways happening. But when there was absence of it, and remember I told you that at that time, it was very, very difficult to isolate and purify just all the proteins because there are thousands of proteins. So maybe when you are purifying, some protein got left behind, or you could not purify it, or some additional things came in, right? So they use digesting enzymes, and using the digestive enzymes to digest DNA specifically, which the enzyme is called DNase, then they saw no transformation. And that was a proof. Okay, is that clear,
Yes, sir.
Yeah. Okay, cool. So in the last class, I told you that we will talk about radioactive labeling. So anyone went back and studied what is radioactive labeling? No worries, Aisha. I hope your network is stable now. Can you hear me? No, our network is not stable. Anyways, yeah. So Iram, Alia, did you go back and looked at radioactive labeling? What does this mean? Anyone? Okay, so we'll discuss about it in a bit. So let's see what was the premise and background of this experiment. So Hershey and Chase, the model, yes,
So the spotlight feature is on.
Sorry, come again?
The spotlight feature is on.
Yes, it is on. Yeah, I was just, yes, it is. So basically, what was the premise of this experiment? What was the model system that they used? Or I should say the organism. That they used was bacteriophages. Now, the name has bacteria in it, but it is not a bacteria. Bacteriophages are viruses, okay? So they are viruses that kill bacteria or infect bacteria, okay? So the word bacterio means bacteria, and the word phage means comes from phage, which means eating. So they eat bacteria, they kill bacteria, and then that's where the term comes from. So these viruses, how do they function is, there is a bacteria, okay, let's say E. coli in this case, okay, and on this bacteria, these viruses they have a head, a neck, and tail-like structures. So they sit on top of the bacteria and inject their genetic material inside. So they drill a hole, literally they drill a hole in the membrane, bacterial membrane, and they release their genetic material inside. Now this genetic material, the bacteria itself will have its own genetic material. Now this genetic material incorporates into the bacterial genetic material. It goes and gets embedded in the bacterial genetic material. And nice to see you, Iram. How are you doing? Great. So yeah, so I was telling you about how this bacteriophage, which is a virus. So this is a bacteriophage, and this is the bacteria E. coli. It sends its genetic material inside. This genetic material goes and gets embedded in the genetic material of the bacteria. And then bacteria does not know that it has been attacked and the genetic material is already embedded. And normally when bacteria replicates itself, like using the machinery of the bacteria, you can replicate the DNA portion of the virus. And this is how virus makes multiple copies of itself when it is inside, when its genetic material is inside. So basically the coat of the bacteria that you are seeing here, this never enters. This just stays outside. So it is enough if only the genetic material of the virus enters into the bacteria, and that is enough to cause infection and to make multiple copies of viruses. Okay, so that's how these bacteriophages work. Now, why they chose bacteriophages or these viruses is because it is very simple a system. So if I draw a bacteriophage virus for you, so it has a hexagonal or 3D hexahedral head. Okay, this is called protein coat. Then it has a tail-like, neck-like structure. And at the bottom, it has these leg-like structures with which it attaches itself to the bacterial membrane. Now, this, whatever is marked in black, is protein in structure. So the whole coat is protein in structure, the head, the neck, the tail. So I divided it to the head, the neck, and the tail. All these three structures are protein structures. And inside, there is the genetic material, which is sitting inside this protein structure. Is it clear? So this is the genetic material, which is the DNA of the virus. Okay. Now, there are only two things in this system, so it becomes very, very easy. So what they did, as we discussed in the last class, they figured out that sulfur is something which is exclusively present in proteins, and phosphorus is something which is exclusively present in DNA. So they radio labeled, yes,
Viruses have RNA as their genetic material.
Yes, not all. Yeah, viruses have both DNA and RNA as their genetic material. In fact, most of the viruses have DNA as their genetic material. The viruses that have RNA as their genetic material are called retroviruses. So these are a group of viruses which are retro means primitive, old, right? And we also believe with evidences that we have now that it was a RNA world in the beginning. So maybe these viruses evolved early. We still do not know very well about evolution of virus. It's a big topic in itself. And I happen to give a talk on evolution of viruses, which is very interesting. We'll talk about it sometime once if we have time going forward in the classes. But yes, viruses can have DNA and can have RNA as both as genetic material. So this particular one has the DNA as genetic material. It's called bacteriophage, and it attacks bacteria. It kills bacteria. So the very unique thing about viruses. Thanks for bringing up this question. It's a good question. So retroviruses only have RNA as genetic material, but viruses are something so unique and amazing as system, because viruses infect everything, but very specifically. So there are viruses that infect bacteria, and they will only infect bacteria. They are called bacteriophages. There are viruses that infect plants, so they are plant viruses. If these come into our body, which they mostly come every time, if you are in a garden or you're walking around and you breathe in, maybe you are breathing millions of viral particles inside your body every time, but nothing happens because they do not recognize us as hosts. So a plant virus cannot affect a human. A bacteriophage cannot affect a human. Similarly, there are specific viruses that infect particular organisms and not human. There are viruses that infect other viruses as well. So this is another interesting thing, that a smaller virus can affect and infect a bigger virus. So this also happens, which is very, very unique. Anyways, so I hope the doubt is clear, right, Iram?
Yes, sir.
Yeah. So about radioactivity. So we can make elements radioactive. That's called radioactive element, and we'll talk about it later, but how to create a radioactive element. But just know that, let's say, if I have a radioactive sulfur and a radioactive phosphorus. So what they did in their experiment is they took these viruses and they cultured it into environment. So they took bacteriophage, normal bacteriophage, and they split the culture in two different groups. One was one was being cultured in a plate or an environment which contained radioactive sulfur. So all source of sulfur that the virus will need, okay, but then it will replicate, it will make multiple copies. And when we culture virus, we know about viruses that they do not behave living outside the host, right? So clearly, when I'm saying that culturing these bacteriophages, we are culturing them in bacteria, right? Providing them with bacteria. So the culture contained radioactive sulfur, one. And in the second case, the culture contained radioactive phosphorus. Okay. So this was the second culture and this was the first culture. Now, what will happen going forward in this culture? After many generations, what will happen? All the viruses will have their proteins labeled, right? So because sulfur is only present in proteins, not in the DNA. So going forward, these viral particles will have, they will multiply and make new particles. The raw material that they need to make proteins will also be sulfur. The sulfur that is present is all radioactive. So these viruses will have their proteins labeled with radioactive sulfur. Does that make sense to you? Everyone, give me a thumbs up if you understand this. Perfect. And going forward in this case, there the viruses will have their DNA labeled, right? With radioactive phosphorus. Okay, cool. So we have two different sets of viruses now. And now the next thing is allow them to infect E. coli bacteria. Okay. And when it will infect the bacteria, then we will just see that what radioactivity we see from the bacterial cells. Do you understand? So
Yes,
this is
sir.
the experiment, which is very, very beautiful, and also gives us unequivocal proof that what is a genetic material. So in this experiment, the first stage will be infection. So what they will do, they will take two E. coli bacteria, like two groups, okay, of E. coli bacteria, and on one side they will use let me edit this a bit. Here, infection stage. So on this side will be radioactive sulfur labeled viruses. So radioactive sulfur is radioactive active. So it radiates radioactive sulfur. And on this side, they allow radioactive phosphorus viruses. Okay. Which is 32 P. So S35 and P32, you can also call this. Now, what will happen in the infection stage? It will simply just sit and infect, right? In this case, when the protein is labeled, we know that the coat will be labeled, right? Because the coat is made up of protein, but inside is the DNA. So DNA is not labeled here. I should not make it red. Let me make it black. Okay, so DNA is inside. In this case, what you will say is it will be the virus will not have any labeling on the protein, but will have the DNA which is sitting inside labeled, right? Do you understand, everyone? Now, as we go forward, in this case, we know that protein coat does not go inside, right? So protein coat will stay out. This is called the phase where DNA has entered inside. So this is the viral DNA and this is the bacterial DNA. And this stage is called blending. So now after infection, what you want is to blend, just mix it vigorously and agitate it vigorously and make everything that is outside the cell stay out and everything that's inside will stay in. So basically to detach whatever is left, to detach this protein coat. And if I draw here also, this is the E. coli. Again, the bacterial coat will stay out. In this case, the DNA will go in. And this is the bacterial DNA. And then the third step is after blending. So these will go away and this will go away. And after blending is centrifugation. So when we centrifuge, what will happen? There will be two things. What is inside in this case is the DNA of the virus, which is now connected to DNA of the bacteria. But there will not be any radioactivity because all the radioactivity will be in the protein, right? Will not see anything glowing in the cell because everything will be out. So these viral particles, the coat protein, which is left outside, is very, very tiny. So when we centrifuge this culture, bacteria being heavy settles at the bottom. So bacterial cells, write down. Bacterial cells settle at bottom. And these particles, they stay in the supernatant. Supernatant is what does not settle, like the liquid. Okay, it's the similar concept. If you put some sand in the water and then mix it well, it will look muddy. But if you centrifuge, all the sand will settle at the bottom because it is heavier and more denser than the liquid, right? Whereas in this case, what will happen is when you will do centrifugation, the E. coli will have the DNA of the virus, which is now and you will have the code of the virus that will stay in the supernatant. So again, this will be in the supernatant, and this will settle at bottom. So what settles at bottom? We call it precipitate. Okay, precipitate and supernatant. So in both the cases, the bacterial cells will precipitate. So if we look here, sorry, yeah. So if you look at the experiment, this is how it is. So we culture the viruses in two different conditions: radioactive sulfur, radioactive phosphorus. In one case, the protein of the virus will get labeled. In another case, the DNA of the virus will get labeled. And since they both are radioactive labels, we can visualize it because they will send active signal, which will glow. So in the experiment, in the first was the infection stage where both the viruses will infect. And sorry, I forgot to make their own DNAs here. So I will just mark everything. Okay, Parker is saying that my voice is cracking. Is my voice also cracking for Aisha and Mariam? Aisha and Mariam, is my voice cracking for you as well? Okay, Parker says it's fine now. Okay, cool. So not cracking for others as well. Okay, so in infection stage, let me mark everything. So this is the virus, bacteriophage. And this is the bacteria, E. coli. And this is the genetic material of bacteria. Okay. Bacterial DNA. And this inside is the viral DNA. Okay. Now infection will happen, then we will separate the two things by blending, and then we will centrifuge. And in the centrifuge, what will happen? If in the tube, you will see, so this is the centrifuge tube. In this tube, what will settle at the bottom will be the bacteria, right? Bacterial cells. And what will stay in the particles will be the viral codes, right? So this is called supernatant. And this at the bottom is called the sediment or precipitate. Precipitate. Okay. Now, in case one, what you will see is you will see the right, because we labeled sulfur. I'm using two different colors here, but it's not like two different colors will be there, but just to demarcate, you will see the glow. You will understand. But here, it will be coming from the supernatant. But in this case, and if the glow is coming from the supernatant, clearly whatever is glowing has not gone inside the bacterial cells, right? That's why it has not sedimented. And therefore, that cannot be the genetic material. So protein in this case, because we labeled sulfur, so protein got labeled and protein stayed in the supernatant. Whereas in this case, you will have same thing and bacterial cells will sediment like this here. And here you will see the signals, the radioactive signal from the sediment. So radioactive signal from bacterial cell. Whereas in this case, you will have radioactive signal from the supernatant. Now, what does this mean? What was marked in the second case? Phosphorus, and phosphorus is in the DNA. And that got inside the bacteria. That's why it sedimented, and we are seeing the signal from the sediment. So this proves that DNA from a virus goes inside the bacteria. And since we know that whatever goes inside the bacteria is the genetic material of the virus, that's how virus replicate. So hence it was a proof that what is the genetic material? DNA is the genetic material. Do you understand, all of you? Do you understand this experiment? And the bacteria they chose was E. coli. Give me a thumbs up if you understood this experiment. Okay, perfect. So write down radioactive bacteriophage viruses, radioactive bacteriophage viruses, yes, Aisha is saying, sir, could you do a small recap? I find it a bit difficult to understand. Okay, perfect. No, simple. I'll do it. Let's start with here. So you understand that bacteriophage are viruses that infect bacterial cells. That part is clear, Aisha? It will be great if you will keep speaking with me.
Yes, sir.
Or yeah. So bacteriophage, they took these virus. It's okay. Iram, are you leaving now or you are back?
I'm back.
Okay, great. Okay, no worries. We just are revising this experiment. We finished it, but Aisha is having some doubt, so we are doing it again. So what they did, I'm just repeating it again. So what they did, they took viruses which are called bacteriophage. The name tells that they kill bacteria, they infect bacteria. Okay, do not get confused by the name. They are not bacteria themselves, they are viruses. And the bacteria they chose was E. coli because these bacteriophage can infect E. coli. So we know that viruses are very simple systems. Okay, these bacteriophages have an outer protein coat, okay, which is black, whatever is made in black here. And inside that head stays their genetic material, which is DNA in this case. Is that clear, Aisha?
Yes, sir.
Now, the whole debate in the field was whether DNA is the genetic material or something else, right? So they got a little convinced actually by what every MacLeod and McCarthy did in the past, but they were trying to find the best way to show it. So they decided to use these viruses because these viruses have just two things, protein and DNA. So they then figured out that protein has, like it was already known, protein has sulfur in it, which is not present in DNA. And DNA as a molecule has phosphorus in it, which is not present in protein. Okay. Then the idea came, they took this virus, they started two different cultures. in one culture, sulfur was radioactive in the culture. So when this virus will replicate and make more copies of itself, it will need sulfur to make its protein, right? And all the sulfur that is available to this colony of viruses is radioactive. So over time, over 10, 50, 100 generations, which is very fast in virus, it will just take few weeks to generate virus with many generations. So after many generations, most of these viruses will have their protein labeled, right, with radioactive sulfur, because they're making more and more protein in the environment. On the other hand, another set of viruses were kept radioactive fossil. Aisha, do you understand that?
Yes, sir.
Yeah. Then simply you do this experiment. You allow these two differently labeled viruses to infect the bacteria. So first stage is infection. So these viruses will infect the bacteria, send their DNA inside. Now when they will send the DNA inside, in this case, the DNA is labeled. In case of phosphorus, in case of sulfur, the protein is labeled. Then after infection, they give the time to infection. My voice is breaking. It says, okay, let me switch off my camera for some time. Is my voice okay? I'll just be back after explaining this. Is it okay now?
Yes, that's okay.
Okay, cool. So after infection is done, they used a blender, like the actual blender that we use in our homes. Yes, ma'am.
So, but it was not breaking.
It's not breaking?
No.
Okay, I think Mariam was saying it's. Mariam, is it okay for you now? If it's not breaking, then I think it's your network, probably. Okay, I'll come back then. Let me know if it breaks. Okay, so after infection, they just blended the whole solution. Blending, what will do is like agitation, it will detach all the viruses that have been attached to the bacteria's cell wall. They will detach because of this blending thing. And after blending, they will centrifuge. So what will happen? Viruses are so small that they will not settle after when we will centrifuge at a particular speed. But bacteria are bigger than viruses, so they will settle first. This is the rule of centrifugation. Whatever is heavier and denser settles first, right? So the idea is that viruses will stay in the supernatant and bacteria will settle at the bottom. Now, since we have already allowed the infection to happen, the genetic material, whatever it is, whether it is protein or it is a DNA, would have entered. And since we have labeled both DNA and protein, we will get to know what has entered inside. Make sense? Do you understand that? Aisha?
Yes. Yes.
Aisha. Yes. Who was having issue? Was it Aisha or Mariam? Wait, I have to check in the chat. It was Aisha, right? So Aisha, do you understand now till this stage?
Yes, sir.
Yeah. So after blending and centrifugation, all they had to do is to check whether radioactive signal is coming from the bacterial cells in case of culture 1, where they labeled sulfur, or in culture 2, where they labeled phosphorus. So if the signal of radioactivity would be coming from the bacterial cells in case of culture 1, it will prove that protein is the genetic material because protein entered the cells. But if it is coming in the case of phosphorus, it will prove that DNA is the genetic material because it's coming from the cells and DNA entered the cells. So they checked and they realized that indeed the radioactive signal was coming from the cells which settled at the bottom in case where phosphorus was labeled, and in case where sulfur was labeled, the signal was not coming from the bacterial cells, it was coming from the supernatant from the liquid. So this proved that protein does not enter inside the bacterial cell from a virus, it stays out. DNA enters inside, and whatever enter inside we know is the genetic material. A virus only sends its genetic material inside, right? So this experiment proved that DNA is the genetic material and not the protein. Do you understand that, Aisha?
Yes.
Okay.
Yes.
Okay, now write down this line. They concluded that bacteria which was infected with viruses that had bacteria which was infected with viruses that had radioactive phosphorus, bracket write DNA, in the bracket write DNA, radioactive phosphorus, which means DNA, were themselves radioactive. This indicates that DNA was the genetic material that passed from this indicate DNA is the genetic material that passed from virus to the bacteria. Okay, and next line, the bacteria that were infected with viruses, having radioactive sulfur, in the brackets write protein, having radioactive sulfur in the bracket write protein, were not radioactive themselves. Were not radioactive. Yes, Parker, I will repeat. Bacteria that were infected with viruses having radioactive sulfur, bacteria that were infected with viruses having radioactive sulfur in the bracket proteins were not radioactive themselves. This indicates that protein did not enter the bacteria from the viruses. This indicates that proteins did not enter the bacteria from the viruses. Okay. Therefore, DNA is the genetic material and not protein. Okay, so this is the proof. Please read this part from NCERT and try to understand what they have written with respect to what we have discussed here. Okay, everyone, this part is clear. Give me a thumbs up. We are on the same page now. We know that DNA is the genetic material and this is how it was figured out. Perfect. Now, before we move forward, let me also talk quickly about RNA, right? We were discussing some things about RNA in the previous classes as well. So tell me, so what was the difference between RNA and DNA quickly? DNA is a double helical structure, right? Whereas RNA is not, right? And what are the other differences?
DNA is less reactive.
DNA is, sorry?
Less reactive.
Yes, that's right. Whereas so DNA is more stable also chemically and structurally than RNA. So all these things we know, but then the question comes that in the central dogma, DNA is present and then it needs to form a RNA to get translated into a protein. So then they asked, where did what came first as the genetic material, a DNA or a RNA? And there are certain viruses also that contain RNA, right? So these retroviruses, tobacco mosaic virus, and there is one bacteriophage also called QB. The QB bacteriophage also has a as the genetic material. RNA both act as genetic material. So which came first is the question, right? Now, first to answer this, let's go through a question which is asked in boards. Write down the question. The question is, what is or what criteria should a molecule fulfill? Should a molecule fulfill in order to be an ideal genetic material? An ideal genetic material. Okay. This question can also come in other ways, like what are the characteristics of an ideal genetic material? Why DNA is an ideal genetic material, but other molecules are not, etc., etc. Okay. So let's talk about it. So first thing that molecule should have is that it should be able to replicate, right? Whatever is the genetic material, able to replicate. Okay. So this question can also come as why DNA is the genetic material and not protein. So write down this also. This also comes in boards many times. Why DNA is the genetic material and not protein? Because Hershey and Chase just proved that DNA is the genetic material. They did not, their experiment did not tell why. So first criteria is that it should be able to replicate. Correct? It makes its copies. So the process of replication comes here. Now, do you think proteins can replicate themselves? A protein can replicate and make more proteins. We know that DNA can replicate itself. In the central dogma, DNA can make more DNA. Can protein replicate and make more protein? Think about it. Think about it. Think about it. Let's go back to central dogma. Where was it? Where was it? Where was it? Oh, was it not here? It was in the last. Okay, last chapter. So we're not going that back. But if you remember the central dogma, you must have it in your copies. A protein can only be translated using a mRNA, right? A RNA. So ribosomes only read RNA and hence make proteins. Is it clear, everyone? Give me a thumbs up if this is clear. Yes. So a protein cannot replicate itself without the help of a RNA, or without it cannot replicate itself simply. Only a RNA can make a protein uh, uh, with the help of ribosome. So ribosome reads the RNA and makes protein. Okay. So this first property is not present in protein. So right here on the first step, protein is discarded. It does not fulfill even the first criteria. So it cannot be the genetic material. So it has to be DNA, right? What is the second thing? If something is acting as a genetic material, tell me, do you want the genetic material to be stable chemically, or you want it to be unstable chemically? Remember that genetic material is used to carry information for each and every feature that we have, and each and every feature that we are going to pass on to our offsprings. So it's like a hard drive where you store all the data regarding project existence of the body. Okay. So what would you like the genetic material to be? Stable or unstable? Logically. It should be stable, right? It should be stable chemically and structurally, both. Because if the genetic material is not stable, our ancestors, gave some gene, that gene changed human being looking like humans. That's problem, right? Like parents had some features like eyes, babies do not have eyes because the gene is not there. So that's a disorder, that's a problem, right? A species cannot exist and continue to evolve if the genetic material is not stable. So the second criteria is the genetic material should be stable. This criteria is fulfilled by proteins. Remember, proteins are also stable, okay, chemically and structurally. But since the first criteria is not fulfilled, they cannot qualify. It's like if you do not, if you do not clear round one, you do not go to round two of the interviews. So protein are out at round one only, even if they were able to qualify round two. You could have said, no, oh, only if my round one was clear, I would have, rocked in the interview round. Many people often say that. But the first thing is, that's why the first round is there. You have to clear the first round, then only. Even if you are good at clearing interviews, it does not make any sense, if you do not clear round one. Same happens for many entrances. So proteins are out right here at the first step. So second step is stable. So now we have two things competing, DNA and RNA. Proteins are gone. So tell me, which is more stable, DNA or RNA? DNA, right? So DNA is more stable, and hence DNA got 1 point up. So DNA 1, RNA 0. DNA is more stable. Okay. Let's look at the third criteria. And coming to replication, RNA also cannot make new RNAs. DNA can make RNA. DNA can make DNA also. RNA can also make DNA through reverse transcription, right? Remember reverse transcription going back, but RNA cannot make RNA. So that's also a problem. Okay. But this is textbook knowledge that we had till now. There are labs in the world, including the lab of Nobel laureate Zostek. So he visited my university last month, in the month of July. Yeah, July only, like May, June. Yeah. And he gave a talk that his lab is now working on figuring out, when RNA was the genetic material in the primitive world, how it used to replicate. So there are research that suggests that maybe back then RNA had the ability to self-replicate. How? It's not very clear, as because today we cannot make RNA self-replicate itself. But research is where we go scientifically and we figure out things which we don't see now. But for now, we will believe that RNA cannot make more RNAs. They cannot self-replicate. DNA can do that. Protein also cannot do that. DNA is more stable than RNA. Protein is also stable, but protein is out on step 1. The third is it should be stable, but it should also give scope of slow changes, right? There should be chances of making changes in that material. If it is stable, but it is very, very stable, where you cannot make any change in it, then it is a problem. Then no evolution will happen, right? So let's say the first human that evolved, or the first set of species that evolved, they have black hair. There is a gene for black hair. Now that gene is set in time, it will never change. Then all humans should have black hair, right? But is that the case? Do all humans have black hair? No, some have grayish, some have brown, some have blonde hairs. Different people have different kind of hair. Even the texture is different, silky, straight, curly, all sorts of, which means the same gene have multiple versions. And now we know what we call them. What we call them? Different versions of the same gene. Come on, people. Chapter 5 was all about it. Alleles, remember? Alleles are different versions of the same gene, right? Capital T, small t. One, the gene is for height. One allele gives you a slightly longer height. The other allele gives you shorter height. Same is the allele which is for the skin color, right? Okay, cool. So it should be able to change slowly through mutations to undergo evolutions. Write down. It should provide a scope for slow changes. And in the bracket, you can write through mutations. Slow changes. Okay. Because this is very important. Also write down another line. This is important for evolution to happen. This is important for evolution to happen. And the fourth criteria that a genetic material should have is it should follow Mendelian rules. If it is a genetic material, whatever Mendel has discovered, it should follow that rule. So write down, it should be able to express itself. It should be able to express itself in the form of Mendelian characters. In the form of Mendelian characters. Okay. In short, you can say it should follow the Mendel's law of inheritance. Okay. If these 4 are fulfilled, then it is a genetic material. Now let's see how many proteins fulfill. Protein cannot replicate, 0. They are stable. Yes, they are stable. So protein fulfill 0.2. They should provide a scope for slow changes. Actually, proteins also do mutate. So let's say they also fulfill 0.3. But they do not express in Mendelian characters. They are not able to express themselves in the form of Mendel characters all the time. Okay. So that's where they lag. So protein only fulfill 2 of the 4 points. So hence they are not good for, they are not an ideal genetic material. Let's see how many RNA fulfills. Does RNA fulfill point 1? No, they cannot replicate. They need DNA. RNA can only be made from DNA, right? Remember, the research, we are not talking about the advanced research as of now. As of now, the evidence is current RNAs cannot replicate. So that's what we know. Okay, always remember that. So RNA does not fulfill point 1. Does it fulfill point 2? Not at all. It is not stable. Does it fulfill point 3? Yes, it mutates. But it mutates it can mutate very fast also. It can mutate slow also. It can mutate fast also. But let's say it fulfills point 3. Does it fulfill point 4? Yes, it can fulfill point 4 in cases where it is the genetic material, like in organisms where this is the genetic material, it can fulfill point 4 as well. Okay, electricity is back. So RNA also fulfills 2 out of 4 points. So RNA and protein both are not good as genetic material, whereas DNA fulfills all the 4, right? Hence, DNA is the ideal genetic material and not RNA or protein. Okay, everyone? So this is what you have to write in exam if they ask you why something is genetic material and why other is not. Okay? Clear to everyone? Give me a thumbs up. If everyone is on same page, let's then go forward. And by going forward, I mean, I'll take you back in history, biological history, and I'll talk about something called RNA world. So we have evidences now to suggest that at one point of time, the biological world had just RNA. Okay, so it was called RNA world. So RNA world hypothesis. So it was given as a hypothesis, but now there are evidences to suggest it. Now, what are the evidences? The evidences, because in science, we do not accept any story as truth. We rigorously ask for proofs. Now, if at one given point of time, there was just RNA, and then RNA had to do more than one roles, right? There was no DNA. Okay, let's say, and it was the first genetic material. Now, if it was the first genetic material, then it must be doing multiple roles, right? Yes or no? Now, are there any proofs where we can see RNA playing these multiple roles? First, do we see anywhere in the world that RNA actually is the only genetic material present and there is no DNA present in any species or any system or anywhere? We just talked about it. Is there a system where there is no DNA and only RNA is present? Yes, there is a system called retroviruses. I just told you, their genetic material is RNA. So they do not have any DNA inside their coat, right? And they're still surviving. So how do they work then? So they work by infecting organisms, those who have DNA. So they also carry an enzyme which converts the RNA back to DNA and then infects the organism and make more copies of the virus. Okay. So retroviruses have an enzyme called reverse transcriptase that helps them to do reverse transcription in central dogma. Okay, that is one proof. But with one proof, we are not very happy. I'm like, if you are saying that RNA was the only material, then it should be able to do jobs like in metabolism. It should also help in metabolism, because if only RNA is there and there is no DNA and no protein, what is catalyzing all these reactions? Okay, so can RNA do multiple roles like catalysis? So we know currently Tell me, class 11th, you have studied metabolism, not metabolism, but you have studied digestion, right? You have also studied metabolism, what I'm saying. You have studied about proteases, lipases, everything, metabolic activities, right? No? Oh, it's in class 12th. Okay. Human
No, sir,
physiology.
it is there. It is
Oh,
there,
it's there, but
it
it was
deleted.
deleted. Oh, that's great. How do you feel now, people? Do you feel happy about? I'm sure that in class 11th, you must have felt happy. Oh, wow, this chapter is deleted. One less headache. Okay, but go back, read that deleted chapter on your own. Believe me, if you will follow what I'm saying, there are more and more chances with everything I say and you follow, there are more and more chances of you clearing the entrances. You need to know this. So in digestion, how do we metabolize substrates? We metabolize substrates by, I'll just keep my video off for 1 minute. We metabolize substrate through enzymes, right? You know enzymes, digestive enzymes secreted by pancreas. Also in the stomach, we have enzymes. So these enzymes do catalysis of a biological reaction. They are called biological catalysts. So when these were not there in the primitive time where it was RNA world, can RNA catalyze a reaction? Can RNA do this? Are there any proofs? Yes. So there are proofs where a particular biological reaction is catalyzed by RNA catalysts and not by protein. Okay. And those are called ribozymes. Write down the two evidences which helps us prove that RNA world hypothesis is correct. First is RNA was the first genetic material. RNA was the first genetic material. And still today, some viruses have RNA as their genetic material. So this is first proof. And still today, some viruses have RNA as their genetic material. Second point, write down. There are also evidence to suggest that important life processes like metabolism and translation. Translation is making of protein. So to make protein, we need RNA. We don't need directly, we just need RNA, right? DNA makes RNA and RNA makes protein. There are evidence that important life processes like metabolism and translation also evolved through RNA. This is the second proof. Third point, write down. There are certain RNA molecules. Third point. There are RNA molecules called as ribozymes. RIBOZYMES. Called as ribozymes that act as RNA catalysts. For some important biochemical reactions. In living organisms. For some important biochemical reactions in living organisms. And these reactions are not something, as a side by side, what do you say, time pass reactions, that if it happens, it happens, if it doesn't happen, does not matter. These are reactions in the human, even in human body, which are very, very important. If they do not happen inside the cell, it will be very, very difficult for humans to survive or develop or do some things. And they happen through RNA catalysts called ribozymes. Okay. So this is another proof that yes, RNA is capable of being the genetic material. RNA is also capable of giving rise to proteins, and RNA is also capable of catalyzing reactions, which means acting like an enzyme itself without any protein enzyme. Does these 3 points make sense to all of you? Give me a thumbs up if you understand all of this, if we are on the same page, everyone. Yes, Mariam says, Aisha, Iram, perfect. So till now, we understood everything that you needed to know about DNA and RNA world hypothesis. So RNA world hypothesis, small paragraph in your NCERT. Please go and read that. Okay, you don't need to know much more than this. What is left now in this chapter is all about how things happen. So basically the central dogma is left, which contains 3 things: replication, transcription, and translation. Okay. So these 3 things we will start in the next class. And the next class I will finish replication, and so first thing that we will do is replication and transcription. For translation, I have to teach you genetic code first. So in the next class, we will do replication and transcription. Replication is DNA to DNA and transcription is DNA to RNA. Is that okay, everyone? And so that we can go fast from here, because now fundamental foundation is done. So in every chapter, I will go slow till the foundation part is there. Once we have understood whatever is needed, we will start pacing up. So from next class, we have to pace up because all the foundation is done. So you understand DNA, RNA, molecules, everything. So read replication, write down your homework. You have to read about replication, which means making DNA from DNA. And just read from NCERT. Don't go to other sources which are more complex and complicated. Read replication and transcription, DNA to RNA. So DNA to DNA and DNA to RNA, we will start in the next class. Okay, everyone? Okay, so I'll see you all in the next class then. Take care. Bye-bye. We'll start the